My name is Rod Richard. This is my critical assignment preparation 1 for ATP Pathways. The assignment asks that we create a diagram to detail it. So I did that. Here's what it looks like. But I also created a PowerPoint to make it a little easier to put all the information down to further explain it. So I'll bring that up now, and we'll go through the presentation. So again, critical assignment preparation for ATP Pathways for KIN 516. One of the ways is through cellular respiration. It's basically the process of turning glucose into ATP. It actually costs 2 ATP to do, but produces 36 in the long run. So definitely worth it. There's a few steps here. Step 1 is glycolysis. The energy is needed to break down the glucose. And so again, like I said, it's definitely worth it because more ATP is going to be made. What happens is we take those 2 ATP and they split the glucose into a couple of carbon-3 pyruvates or pyruvic acid. But from that, 4 ATP are produced as well as NADH. Going back, that all kind of happens in the cytoplasm. From there, that pyruvic acid is again broken down and to produce more NADH and FADH, which are transported through the electron transport chain. There, the carbon bonds with 2 oxygens and becomes carbon dioxide, which is picked up by the blood and released to respiration. So this Krebs cycle here is all kind of happening during and inside of the mitochondria. A lot of things going on. There's some citric acid. There's 5 carbon compounds. You see there's the hydrogen and carbon dioxide kind of coming off at different points. So from this, we get an additional 6 ATPs. Step 3, as I mentioned, the NADH and FADH are sent to the sent to the electron transport chain. And so this is what that looks like in greater detail or greater depth. So you see the NADH coming in here and FADH2 coming in here. There's a lot of hydrogen ions out here. And so oxygen enters into the membrane of the mitochondria through respiration. And the hydrogens on the NADH and FADH carry energy into the cell via the gradient. So that's this area here. And the hydrogen is then released from its carriers into the and bonds with oxygen to form water. Right, and that water is released as waste. These pictures are kind of giving us an example of what's happening inside the cell to create more energy as the body needs it. Those 3 steps are the basis of it. If it were to be a fat molecule, it'd be a little different. You still kind of have the same setup, except now we have a fat molecule, which is triglyceride, being split into fatty acids and glycerol. That glycerol now is if the ATP levels are high, it's going to be converted into glucose, right? And so it's going to go up here and it'll be saved and wait until we need to use it. But if the ATP levels are low, it's going to be converted into pyruvic acid, just like the glucose from the previous example. And from there, it's going to enter the Krebs cycle. Be converted into acetyl coenzyme A, and then into a Krebs cycle. As you see, kind of written here, but you get to see it here. And that's it. Everything pretty much in a nutshell. There's obviously quite a bit that goes into it. It's something that's very, very specific to each person's body, very technical. But that gives us a pretty good example of what happens there. Again, glucose through glycolysis in the cytoplasm is broken down into two pyruvic acid compounds. That it turns into acetylcoenzyme A, sent into the Krebs cycle. A bunch of things are thrown off. 2 carbon dioxides, 4 carbon dioxides, 2 ATPs, some NADH here, some FADH2. This is going to enter into the electron transport chain, which is going to be basically phosphorylation. And our end point, now we have a total of 32 ATPs, which we started from using 2 ATPs.